DNA. In this module, you will learn about the structure of DNA. DNA, that is deoxyribonucleic acid, is one of the most important biomolecules. DNA is a polynucleotide, which means that it is made of a large number of nucleotides. A single nucleotide in turn is made of three components: a sugar, a nitrogenous base and a phosphate group. The sugar in a nucleotide of DNA is beta-D2-deoxyribose, which is a pentose with 5 carbon atoms. The nitrogenous base, on the other hand, is a nitrogen-containing heterocyclic compound that can be adenine, guanine, cytosine and thymine. When any of these nitrogenous bases attaches to the first carbon of the sugar, a nucleoside is formed. To a nucleoside, a phosphate group adds to the 5th carbon of the sugar to form a nucleotide. This way, the 3 components of a nucleotide are united. Now, all the nucleotides of DNA have the pentose and the phosphate group in common, but they may differ in their nitrogenous base. As there are 4 different bases for DNA, there are 4 types of nucleotides in DNA. Any 2 nucleotides of DNA, similar or different, can join together. They do so by forming bond between the 3rd carbon of the pentose of one nucleotide and the phosphate group attached to the 5th carbon of the pentose of the other nucleotide. The structure resulting from the union of 2 nucleotides is called a dinucleotide, and the linkage between the 3rd carbon of pentose of one nucleotide and 5th carbon of the pentose of the other nucleotide through a phosphate group is called a phosphodiester bond. By forming similar phosphodiester bonds, more nucleotides add to the dinucleotide, forming trinucleotide, then tetranucleotide, and so on. Gradually, a long chain of nucleotides or a polynucleotide chain is formed. This long chain of nucleotides is also called a strand of DNA. A strand of DNA, as you see, has a sugar-phosphate backbone to which nitrogenous bases are arranged. The sequence of nitrogenous bases in a single strand of DNA is the primary structure of the DNA. Now, the nitrogenous bases of one strand of DNA join the nitrogenous bases of the other strand by forming hydrogen bonds. Interestingly, cytosine of one strand always joins the guanine of the other by 3 hydrogen bonds, and adenine of one strand joins to thymine of the other by 2 hydrogen bonds. Since the bases of one DNA strand always have the complementary bases in the other strand, the base pairing between 2 strands is called the complementary base pairing. This complementary base pairing between 2 strands of DNA is the secondary structure of DNA. The 2 strands of DNA that are held together by hydrogen bonds spiral around each other, forming a double helical structure. This structure, having 2 DNA strands wound around each other, is the tertiary structure of DNA, which is also called double-strand helix of DNA. Let's recap. DNA is a polynucleotide. A nucleotide of DNA contains beta-D2-deoxyribose sugar, a phosphate group, a nitrogenous base that can be adenine, guanine, cytosine or thymine. Different nucleotides of DNA join by a phosphodiester bond to form a polynucleotide chain or a strand of DNA. The nitrogen bases of one DNA strand join with complementary bases of another strand by hydrogen bonding. The 2 strands of DNA that are held together by hydrogen bonds spiral around each other, forming the double-strand helix of DNA.